Hey guys, welcome to Unit 2 of the AP Bio Unit Overview. And today we'll be going over Unit 2, which is Cell Structure and Functions. And to understand that, we have to first understand the differences between eukaryotes and prokaryotes. And so, firstly, most eukaryotes are multicellular, meaning they are made up of multiple cells, whereas prokaryotes are mostly unicellular, meaning they have only one cell. Next, uh, all eukaryotes contain a nuclear membrane surrounding the nucleus, whereas prokaryotes lack this nuclear membrane, so it is absent. Other organelles such as Golgi bodies and mitochondria are absent here. They're present in eukaryotes. Whereas they are once again absent in unicellular prokaryotes. And then we have cilia and flagella, which are present in prokaryotes, which are basically like the tails for the cell, and they are absent in eukaryotes. So now let's go to the structure and talk about the structure. So let's start here. So we have a Golgi body, which essentially modifies and matures the protein made by the rough endoplasmic reticulum. So think of it like this: it receives a package, it modifies it into more useful proteins, and that is then sent out. Either out of the cell or inside the cell in packages called vesicles. Vesicles. Ribosomes manufacture proteins. They are the protein house of the cell. Smooth endoplasmic reticulum manufactures lipids and breaks down some waste. Then we have the mitochondria, which by now you should know is the energy house or the powerhouse of the cell. Then we have the lysosomes, which are also the digestive center for the cell. They have digestive enzymes which are secreted into the cell to digest wastes. Then we have the nuclear membrane, which protects the nucleus from being degraded by enzymes inside the cell. So it basically surrounds the nucleus. And nucleus is where we find the nucleolus, which contains the genetic information packed in packages called chromosomes. Then we have the rough endoplasmic reticulum. And rough endoplasmic reticulums are basically just smooth endoplasmic reticulum which have ribosomes. These are ribosomes. They have ribosomes attached to it. So once an mRNA goes out of the nucleus and tells the rough endoplasmic reticulum to manufacture the protein, it gets the signal and it starts manufacturing proteins, which are then sent to the Golgi bodies. And cell membrane oversees the traffic that is coming in and out of the cell, monitors traffic. Vacuole basically stores some sort of food or energy, and centrosomes are important in mitosis and meiosis, which is important in cell division. So, meiosis. These are basically all of the cell organelles summed up. Then we have active and passive transport. So, to understand active transport, you have to understand this. For example, if I'm keeping a, a container which has water filled, and I have a hole here, and the other container is also the same size. It is the same size, but it has less water. Let me make it a bit neater. So, one container which has two compartments, and in the middle they have a hole, and the one part of the container is filled up till here, whereas the other part is filled until here. What do you think will happen? The water will move from a higher concentration, it'll go this side, and will keep going until these are balanced, so the water is leveled off in both the containers, and that is basically active transport summed up. And active transport is important in cell membranes as it allows small non-polar atoms to move across the cell membrane without any help. So these would include oxygen, carbon dioxide, and etc. Then we have the small and polar, and these include, one of which includes water. If you remember from the previous video, water has a slight charge. Water has a slight charge on its hydrogen and oxygen. So this would be negative, this will be positive, and this will be positive. So in order to combat that, and let me remind you, polar molecules cannot pass through the cell membrane because the inner part comprises of hydrophobic tails, which are basically lipids, phospholipids, which have two lipid chains attached, and so the inside, it basically repels all this polar molecules. So in order to go through, we have a channel protein which allows water to flow from high concentration to low concentration in this direction, since there are more particles this side. And this is basically like a tunnel through which the water molecules go through. And active transport also works in a similar fashion, which is like a tunnel that helps the particles go through. But active transport requires ATP, or in other words, energy, since you are pumping water the other way. So for example, let's take the same example. We have a container which is basically like this. It has a small hole in between, and water is in the same scenario, meaning one is filled more and one is filled less. And passive transport would basically be high water is going downwards, which means to a lower potential. But in active transport, water actually goes the opposite direction, and in order to combat gravity and to go upwards, it needs some input, and that will always be possible only in the form of energy. In this scenario, the water will be filled and there will be no water molecules here. And the mechanism in active transport is similar in cells. So we have less particles on this side, we have more particles this side, and we need these particles to we need these particles inside the cell, but our cell already has a lot of it, but it still needs more. So what the channel protein does is it takes an input of energy, it opens it tunnels and allows a particle or multiple in some cases to go in, and then it changes its shape so that this protein could go into the cell. This could also be the case if this is outside the cell and the cell is secreting waste. One of the example of active transport is in the form of the Na plus K plus pump, sodium potassium pump. So in this, what happens is basically we have a membrane, which I'll just make a one line just to make it neater, and there's a channel protein here, and this is the outside of the cell, and this is the inside. So a cell often has Na as waste, so it will have a lot of Na here. Let me just make it clear. It has a lot of Na here and very few here. And on the outside we have a lot of potassium. We have little potassium, and on the inside we have a lot of potassium because we need it inside our cells. So what the protein does is it takes in three sodium ions, it throws it away, it throws it outside the cell, and then once the Na ions are removed from the cell, it takes in two potassium ions and gets them inside the cell, and that is basically the sodium potassium pump. Some other important points in this chapter is the negative ICRT formula, which is used to find out the water potential. This will be provided to you in the AP data booklet, so you won't have to memorize this formula, but you have to be wary of the negative sign, since many people including me get confused on this sign. More types of transport includes endocytosis and exocytosis. And endocytosis, you can memorize this by thinking of endo as in, meaning some stuff or organelles or food or something is being transported into the cell. So and how this occurs is in a very cool fashion. So let's take for example, this is some sort of food and this is a cell. To engulf this food particle, the cell will need to have its arm around this food, and slowly and steadily it will slowly engulf this food particle in a vesicle, and this is phagocytosis. Phago means food, means it is transporting some sort of solid inside. And in penocytosis, it is water, water or some sort of liquid which is being transported inside. And then we have exocytosis. Exocytosis is basically the removal or secretion of waste. So, for example, we have a vesicle which is carrying waste inside. So, what happens is this is found inside the cell, and after some time, this is secreted out in this manner, and then after some time, it will be secreted out of the cell. So, this is a pretty general overview of the whole topic. And if you have any doubts or any questions regarding any of the material discussed in this chapter, feel free to leave a comment. And till next time, I'll see ya. Have fun.